Hello, hola, good afternoon, buenas tardes, Arratsaldeon. We are talking from the center of Navarra in Pamplona, from one of the home bases of the IGEM in the planetarium. And we will be talking about a very exciting project, Genome Right project. And we have the key leaders of this initiative, and I will introduce them briefly to let them speak. We will first have with us Amy Keynes-Watts. She's the general manager, the coordinator of the Genome Project Right, and she will tell us about how we can join this global community, and how can we join also the consortium of companies that are making this happen. So Amy will go first. Then we will have again with us Andrew Hessel. I was really lucky to meet him many years ago in the States, but he was with us at the very moment that the paper, the foundation paper for Genome Project Right went live. So I remember that time here in the planetarium with you, Andrew, and I'm very happy that we are still again connected. I look forward reading your upcoming book, so maybe you will tell us about that a little bit. And then we will have a presentation and a talk with Dr. Church. George Church is one of the main leaders in the world in genetics, synthetic biology, and science in general. I'm really an admirer, and I am very humbled to be here with you. So I just stop talking. We are in Biospain, we are in Navarra, Pamplona, and we're trying to develop a local community that is part of the network driving the new bioeconomy. And with your help, that's fun and easy. So go ahead, Amy. Thank you.
Thank you so much, Juano, for the kind introduction. I'll go ahead and share my screen. Good morning and afternoon, everyone. Thank you for inviting us to speak today at your conference. We're honored to be here. So I'll get started and frame the organization for you, introduce you to our consortium, and then Andrew will follow, and George will get into the details on the science and technology of GP Right. Genome Project Right, designing genomes for a better future. GP Right is a global consortium focused on engineering biology. The consortium.
You're missing your slides.
Oh, you can't see my slides.
We can.
It says I am sharing.
Now it's there. Thank you.
Okay. Should I start over? Okay, this is basically the first slide. My apologies. GP Right is an international consortium focused on genome engineering. As you can see from the image here, it's international in scope, includes approximately 2,000 members across 200 institutions and companies. We're currently in 17 countries on 5 continents, and we're thrilled to have Spain joining our consortium as one of the newest and most recent countries to join our team. We're rapidly expanding global outreach and have recently formed an industrial advisory board to really oversee the launch of our technology thrust. We have a number of industrial affiliates, as well as 9 interdisciplinary working groups. The GP Right leadership team includes a board of directors and scientific executive committee. These are real pioneers in genomics and synthetic biology. So it's a fantastic international team. Our industrial advisory board includes the members listed here, and we have a number of industrial affiliates throughout our consortium as well. GP Right's mission, the goals are scientific and technological, and an overarching ethical framework guides all aspects of GP Right projects. Scientifically, the goals are really to understand the blueprint of life that was provided by the Human Genome Project. And we do this through a number of pilot projects, as well as through our international community project, which George will be discussing in greater detail. The overarching goal is really to drive transformative advances in medicine and environmental health. Technologically, the goal is to reduce the cost of engineering and testing large genomes in cell lines 1,000-fold over the next decade. GP Wright's roots stem from the Human Genome Project, and here I'm just illustrating the key milestones that flowed from the completion of that first read of the human genome. By contrast, you can see that genome writing is really lagging behind genome reading because of the inefficiencies and cost that are still associated with genome synthesis capabilities. And now is really the time for genome writing. It's the integration of the reading and writing technologies that makes GP Write possible, where we're looking to drive transformative advances across many sectors of society, from medicine to food and agriculture, to energy supply, chemical industry, as well as developing next generation materials. So where the state of the art is really limited to editing single genes, GP Write is looking to advance these capabilities to the network level and then the whole genome level. So how are we doing this? GP Write science is, the community project is focused on recoding a human cell line, an induced pluripotent stem cell line. And again, George will discuss this in greater depth, but by recoding a human cell line, you can endow it with new programmable biological functions, such as virus resistance, endowing cancer resistance, improved genomic stability, and this creates safe and versatile human cell lines for very valuable biomanufacturing applications. On the technology side, our tech team actually published a roadmap and white paper about 2 to 3 years ago that nicely set forth the key milestones that we need to achieve over the next decade to really reach our goal of driving down the cost of writing large genomes 1,000-fold over the next decade. And the first milestone that they identified was a next generation computer aided design software platform. And so that's how we've launched our GP Write tech effort. It's focused on creating a next generation CAD. That first version is going to be rolled out at our upcoming conference, which we're very excited about. And it will be a living CAD so that we will be continuously advancing it and keeping it sort of cutting edge. Advancing DNA synthesis capabilities, genome editing capabilities, chromosome design and construction, as well as capabilities to deliver designed chromosomes into cells will also be a key effort, and overall automation of genome engineering technologies. So how do we plan to achieve these goals? We've envisioned a GP Write hub. This includes a virtual foundry and incubator where the GP Write CAD is a foundational technology. Thinking really about the CAD as sort of a one-stop shop where you can design a genome, and participating members of our industrial consortium and board will have extensibility built in so that you can order directly their products and services, which can then be shipped to a participating biofoundry for assembly and testing and returned to the original designer for phenotypic assays and testing in the lab. The GP Write CAD software. So just a bit about the technology itself. We're partnering with Lattice Automation in Boston, Massachusetts, and they're helping to actually to build the platform. It will be the first whole genome design tool for rapid and automated design of synthetic genomes. The way that it works is you input a desired genome, apply a series of directives, editing directives, and then there's systematic checks for known pathogens and toxins. This is an image of the graphical user interface. So the goal here was really to make a very user-friendly GUI so that you don't necessarily have to have a computer science background to use the GP Write CAD. The goal is really to democratize the field of genome engineering and make it much more broadly accessible. So genome visualization, there'll be sequence level visualization available from the CAD, visual comparison of the original and modified genome, graphical visualization of directives, as well as abstract design representations and the ability to view design history and cross-compare. Genome design, you can apply dynamic genomic edits, global or individual codon swaps. You can apply comments as annotations to accompany the directives. There'll be capability to apply genomic watermarking, edit, insert and deletion directives, as well as user warnings related to genomic conflicts. So if you've designed a sequence that may not be compatible with another sequence in the genome, there'll be a warning that's issued, and also the capability to apply codon optimization principles. The genome output. So here there'll be a FASTA file output, a visualization of wild versus the modified genome, and segmentation and or order forms to partner and build extensibility with our industrial partners. Encoding safeguards, we're going to partner with the International Gene Synthesis Consortium to really make sure that we're out in front of any biosecurity concerns. We're also, as this is an emerging field, thinking a bit more broadly and creatively about other biosafety mechanisms that we can endow in this CAD platform to ensure that GP Right is really out in front in terms of talking about this and engaging the community more broadly on the topic of biosecurity. And this is just a nice visual infographic which illustrates the CAD platform as a foundational technology in this virtual foundry and incubator that we're building this international ecosystem, GP Right's design, build, test, learn cycle of its own. Initial pilot projects. So some initial pilot projects for the CAD have been discussed, although they're still being finalized, but ultra-safe cells, our recoding effort might be an initial sort of beta test for the GP Right CAD. This project could include hundreds to thousands and even up to tens of thousands of edits across the human genome. So it's a very ambitious project and would certainly put the CAD to the test. So we'll see if we're able to trial that in the near term. Building and designing synthetic chromosomes. So it's sort of a high level topic, but certainly one that could be tested on the CAD. Developing designs of therapeutics for rare genetic diseases, possibly starting with Charcot-Marie-Tooth disease, and advancing delivery systems using split vector AAVs and mini genes that will be designed so that we really build libraries that can be a broadly accessible resource to patients, families, philanthropies, foundations, funding agencies, and biopharma to participate in advancing R&D pipelines for rare genetic diseases. Ways to engage. We invite you to join the GP Right Consortium. If you're interested, please do contact me. You can also, if you're interested, join the Industrial Advisory Board and participate in building the GP Right CAD and contributing to future features that we'll endow. You could join us at our upcoming annual conference, and I'll provide a bit more information here as well, and join the GP Right Consortium. There are no membership fees. So if you're interested, please do reach out after this panel. Please do join us at our upcoming conference on October 21st to 22nd. I'll include a link in the chat. We're featuring keynote presenters, Dr. Church here today, Dr. Doudna, Dr. LaProust, and Dr. Liu. And contact information is provided here. You can reach me at amy@engineeringbiologycenter.org, our website, and information about registering for our conference is below. Thank you so much. It was an honor to be here today, and I will turn it over to Andrew.
Terrific. Thank you so much, Amy. That's a great overview. I'll just start sharing my screen. Just one moment. So screen sharing failed to start. That's always interesting. 1 second. Great. Can everyone see this okay?
It's perfect, Andrew. Go ahead.
Wonderful. So again, Juanjo, thank you very much for the invitation to present today. It's great to see you again. As you mentioned earlier, Spain has been involved in learning about genome engineering and synthetic biology for quite a while. We've been one of the first movers. One of the things I always found really fascinating is kind of the similarities in some of the architectures between one of my loves, computers, and my other love, biology. They're a hierarchical, both share hierarchical architectures, although built with very different materials and operating in very different ways. But I find conceptually today, many people who, because we've grown up with computers, understand the idea of components and circuits and chips and ultimately computers and networks very intuitively, whereas biology is still quite unfamiliar. And even though they share this similar architecture, which I find useful for communicating, we need to appreciate there's some really gigantic differences. Computers ultimately process bits of information. Biology processes atoms. Their computers are built on a silicon architecture. Carbon is the foundation of life. Computers don't self-manufacture, but biology does. Biology operates in a wet environment. And of course, our computers don't tend to care for that. And the logical operations are very different as well. But this particular architecture, I find, is compelling and can give us some indications of how the field might actually advance. Under this framework, a virus would, which doesn't have a complete computing system architecture, is more like a USB stick in search of a processor. Ultimately, though, it's DNA software that that connects all life on this planet. It's a universal operating system, so to speak, for our cellular systems. And I find that it's just remarkable that we can get so much diversity in living systems really just by having a common language. And I was always fascinated by this, by the DNA molecule, as many people are. And I tend to view it this way, as genomics has started to flourish, it started with reading DNA. And where we could isolate the DNA of different organisms, ultimately digitize that code and turn it into a computer file. The reverse process, writing DNA, is what the Genome Project is focusing on, but it's still very early days. When I started getting into science 30 years ago, the most we could actually produce by starting to write our DNA code was just short strings of amino acids, ultimately making proteins. As our DNA synthesis technologies got better, we were able to produce metabolic pathways, essentially the equivalent of metabolic circuits. And now the technologies are getting to the point where we can now rewrite the genome of the organism and boot it up. We've done this for numerous viruses and bacteria today, and we're starting to advance into more complex organisms. And of course, once you can close this loop and go from an organism's genome to a computer file, and then from a computer file back to the original organism, we can now have the ability to fully engineer that organism down to the bit level, which is pretty exciting. These capabilities haven't appeared overnight. It's really the efforts of thousands of researchers and groups over decades. Pre-2000, it was a challenge to write just a few thousand base pairs of DNA code. Over the next decade, we increased that capability to synthesize and assemble DNA 2 orders of magnitude, from from 10K to almost 1 million base pairs of genetic code assembled accurately. And ultimately, this allowed us to synthesize the genome of the first bacterium in 2010. Over the last decade, these technologies have matured and advanced by another order of magnitude. And now the state of the art is writing genomes in the 10 million base pair range. But that's really pushing the limits at this point. But as new technologies are developed, and whether it's the CAD tools that Amy talked about or better synthesizers, we're moving into a future where we're going to be able to engineer larger and larger chromosomes. The largest human chromosome is 250 million base pairs, for example. And once we can start to write larger and larger chromosomes, it really opens up the engineering of more complex species, and ultimately into species that could never have existed without the technologies of genome writing. So looking forward, I believe that, like computers, our bio tools, our tool sets for doing these types of manipulation are going to get smaller, faster, cheaper, and better. We've already seen this evidence across the board with automation and better tooling. Life operates on a molecular scale, so do modern semiconductor systems. And over time, these two architectures really do look to intersect. I started to see this personally over a decade ago with the development of biochips like these ones used in ion torrent sequencers, where the DNA sequencing reactions were really happening on a very specialized computer chip. The outputs of this type of engineering, where we're starting to build organisms, really look to touch everything and everyone over time. And 2020 was really a breakthrough because the COVID vaccines that were developed by Moderna and BioNTech are really one of the examples, not of writing genomes, but of using designer nucleic acids to create a medicine and ultimately turn our bodies into manufacturing plants. This is now these technologies are going to reach billions of people over the next few years. So this is really kind of a turning point moment for the whole field of synthetic biology. I believe the field is going to evolve faster than most people expect because the actual hardware is universal. The cells are everywhere. There's no shortage of these machines, and they're incredibly advanced. They'll execute the code that we write. So really, in some ways, the rate limiting step is just our ability to learn how to program these incredible machines, and ultimately doing secondary things like navigating the regulatory processes to bring products to market and scaling to meet demand. No one is late to this technology at this point. In fact, even though it's been evolving over the last decade, it's just 10 days ago that kind of one of the bellwethers of the field, Ginkgo Bioworks, had their IPO in New York. And really, it's I think this is one of the companies to really look to and learn from how to be successful. But I know that this field is going to be important because kids get it. The iGEM, International Genetically Engineered Machines competition, which has brought together youth from around the world, mentored them and guided them through the technology and human practices, has been incredibly successful and didn't slow down one bit by going virtual last year. And even my own daughter, who was born the same year that GP Wright was founded, takes to these technologies naturally, understands what I'm talking about more than my wife does. And I'm always delighted that the kids' books and materials really get and feature some of the people and technologies that are appearing around us in the world today. And I love the fact that we're already starting to apply many of the things that we're trying to do with GP Write in ways that are accessible to kids. For example, this is a bioart project that we did recently that has genetically engineered bacteria that produce color. It's actually produced using an online fab that applies the bacteria to a growth plate and is fully programmable even by a 6-year-old. So the things will keep changing. And just to close, if you're really interested in this technology, I want to point you to an incredible report. I think it was provided in some of the materials or links about nature co-design produced by Boston Consulting Group and Hello Tomorrow. It's really a wonderful overview of some of the revolution in the making. And then my own book, written with a quantitative futurist by the name of Amy Webb, will come out next year. It's called The Genesis Machine, and it's meant to be an introduction to some of these technologies and helping people realize that they need to be part of this discussion moving forward. And with that, I'll just say thank you so much for giving me the opportunity to present. I guess, George, it's on to you now. Thank you.
Okay. Thank you. Everybody see the slides? Hear me? No? Okay. So I'm just going to focus on one. Andrew and Amy set up a great broad view of what we're doing. I'm going to give a slightly more narrow view of 3 projects, mainly one, which is ultra-safe cells. I just want to thank our collaborators, and any suggestions that you have and new collaborations are welcome. That's kind of the point of this kind of meeting, is to encourage that. Part of the reason that this is so optimistic, that the picture that Andrew and Amy painted is so optimistic, is because we have these factors of 10 arriving sometimes within a year, but definitely faster than the compute revolution, which is already breathtaking. So these are both reading and writing have gone up by over a millionfold to 60 millionfold, depending how you count, largely due to multiplexing. But assembly and testing have lagged a little bit behind. I think that's what's limiting us now more than synthesis and sequencing. There are many, one of the other things to do with GP Write is develop new technologies. And these are include editing technologies. If they're done on a massive enough scale, they're the equivalent of writing. But also epigenetic technologies. So we can show that we can test that we have the full capabilities we want in multiple cell types before we test it out in a whole organism or in a clinical setting. These are some of the startups that I've been involved in recently that have something to do with this project. I'm going to give one slide each for the top 2 on this list. This is my short list of projects where, from a functional standpoint, we think we need very extensive genome-wide edits, or genome writing, let's call it. It's important to focus both on the technical capabilities and the possible applications, because it's a little dance they do. So the 3 that I came up with, or that our team has come up with over the years, is being able to transplant organs between species, the de-extinction of genes or genomes, and this isn't necessarily just frozen genomes. This can go all the way back through phylogenetic comparisons back to the dawn of time, and this has been done for many years. Genes, genomes, viral genomes, so forth. And then finally, the big one is recoding the genome for resistance to all viruses, not just a few. So here's the xenotransplantation. We've now successfully made 42 edits in the germline which shows that we can do extensive editing and still have complete functionality. In fact, enhanced functionality. These are enhanced with respect to their immune compatibility with humans. So these are essentially humanized pigs. They have all these changes here. This is a complete list of pretty much the wish list of the entire field that accumulated over 20 years. And most of it was done in these 4 years in these papers down below. One of the big ones was eliminating all the endogenous retroviruses. And we did this not by just deleting them, but by finessing, knocking out just the polymerase function and not the membrane-bound envelope function. And that turned out to be very significant for 2 different reasons. These organs from these pigs are already in 3 hospitals in preclinical trials. Jim Markman is running the closest one, which is Massachusetts General Hospital. And these 2 startups have been involved, Eugenesis and Kihan, thanks to Luhan Yang, who was a grad student postdoc in my lab and is now in Hangzhou running CEO of Kihan. The second example of 3 categories is this de-extinction, which I think I don't need to talk about. Has been in the news quite a bit, but it's not really about de-extinction of a species, but de-extinction of individual genes mixed with other synthetic biology tools to make something that's neither elephant nor mammoth nor even a hybrid, but much more than that. And it's focused on endangered species that are still alive and on carbon sequestration. So the big one, the last of these 3, is recoding, codon recoding for resistance to all viruses. This is work we have many people from all over the world, but Yuting Chen is from Shenzhen. Ariana Hosoli is at Colossal, the elephant company, and Farron Isaacs is from Yale, and others. And here are some of the things that we published in our science papers in 2016 and 2018. And these include not just viral resistance, but we know how to make cancer resistance, prion resistance. I already showed endogenous retroviruses can be eliminated in pigs. The pig project de-risks a lot of this stuff. Immunodiversity, radiation resistance, senescence resistance, and the list goes on. Some of these involve a small number of edits. As far as we know, some might involve quite a few. Virus resistance, we think, is the largest one so far. We can do many edits at once. In a special case, we can edit repetitive elements, which has been the dark matter that's been hard to read as well as write, but we can now, we're doing that now, and these repetitive elements are involved in interesting phenomenon of senescence, neurogenesis, cancer, and inflammation. So here's our record so far for editing repetitive elements. These are the human line elements in the same iPS cells that we are using for the recoding project. So again, this de-risks at least part of the multiplex editing if we will do it by multiplex editing or some combination with writing, as we did in the pigs. Here we have 22,000 edits where we got a clone. It's a stable iPS human clone that has 22,000 edits. And then here's just quick slides that show how we have shown multivirus resistance already in a microorganism. We're trying to extend it to a variety of eukaryotes, plants and animals, particular humans, cells that could be used in possibly and even in a clinical setting. So we changed one codon, removed one codon effectively genome-wide, and got 3 new functionalities: new amino acids, biocontainment, and this multivirus resistance. And this is the work of a number of teams over the years, starting with Harris Wang and Nili Ostrov recently. In red is the first codon we took out. In green, 2 more from the Chen lab. We're very grateful to their sharing via the literature. And in the blue ones are 6 more that we've synthesized, but have not finished assembly yet. Both the single and the triple have been shown to be multivirus resistant. Here's an example of some early work from our collaborator and ex-postdoc Farron Isaac's lab, where input virus at up to 1 trillion plaque-forming units per mil, really high levels of virus, go down to 0. That's what all these zeros are on the baseline. The only viruses that are not, that manage to make it through are viruses where the UAG stop codon is not critical to their life. And we expected that. So that's why we're going for additional codons, 2 or 6 more. We also have had progress recently in using an interesting combination, machine learning and large libraries. So neither one alone, but together we can do protein and genome engineering with some tools complementary to the ones that Amy mentioned. And this 3 companies were spun off from this machine learning 4 machine learning papers: Dino, Manifold, and Nabla. And then finally, we want to make sure that we can test this in multiple cell types before we commit to either an organism or a tissue in clinical setting. We have, we think, all of the expression clone for at least every one of the open reading frames look like they might be a DNA binding protein. And we have 290 cell type recipes so far. We don't know how many cell types there are yet until these cell atlases are done, but we're trying to keep up with the cell atlases. Alex and Peristu did this work as postdocs, and now they started a company, GC Therapeutics. So we want to invite you to, this is our website that's maintained for the community, specifically on the ultra-safe virus-proof cells. Here's the website if you want it, or you can just ask. And this page goes on and on. It's not, this is just the top part of it. So we invite you for that. Thank you. I'll stop sharing and open it up for questions for all of us, for all of the people here.
Thank you very much, George, Amy, Andrew. It was awesome. I wish we had more time with you, and I really dream that the next time we will be physically together in Pamplona or in Boston or in California. I'm surrounded by some people that came from Europe, different countries, to join us here in Pamplona, and they have plenty of questions. So let's go with some questions.
What have you planned in order to make all this information more worldwide, in order to get known to every people?
So this is one of the key players in our new BioGalaxy iGEM team.
Well, we've haven't exactly been hidden. I mean, there's a fair amount of press both around GP Wright and some of the sub projects. But we're open to suggestions about ways to make this more noticeable, more inviting. We really are very, we have, as Amy mentioned, 105, I think, groups that have expressed some interest in our members, but we're open to suggestions.
Hi. My name is Marjolein Kroijmans. I'm from the Netherlands. I'm here as the co-chair of iGEM Epic. We got invited by Juanjo to come here as part of the social innovation. So thank you so much for inviting us. iGEM Epic is the entrepreneurial program of iGEM. So for iGEM alumni, if they're interested in turning their project into startups, we're here to help you. So my question is regarding the future. We've seen a lot of acceleration, faster computers, faster programs, faster predictions. Where do you see your own fields going in like the next 5 to 10 years? And thank you so much for your introductions, by the way.
Well, I don't see anything slowing down in our, from time to time, people predict slowing down of the Moore's law, specifically for tighter packing of transistors. Right now they're 3 nanometers, which is getting pretty close to a theoretical limit, but with synthetic biology, I think reading and writing genomes, I think we have at least 5 orders of magnitude left before we need to worry about plateauing.
I'll just add that people don't think exponentially in a natural way. Many exponential technologies, whether it's computing or the scaling that we've seen and cost reductions with things like solar cells, have been completely misjudged by most analyst communities. And so I think this is, I put in the slide deck that I think people are underestimating how quickly this will evolve. And again, because it's really limited by our ability to code and compile. The cells are willing to do what we want to program, what we can program them to do. So I think it's going to continue accelerating for a while.
I would just add also here, I can speak to the CAD effort. I can't say how many orders of magnitude we have sort of to go on that front, but I know there's a ways to go in terms of advancing the deep learning and really learning how to map the phenotype to genotype that's being designed. And so we're in the early stages of really sort of advancing the deep learning on that front. And then just to follow up on George's point, yes, we are certainly open to suggestions as to how to gain greater visibility for GP Write. If the iGEM teams are not aware of our consortium, that's our bad, our fault. So please let us know how we can become more visible to your group. I know that social media is an important mechanism, and we have LinkedIn feeds, but happy to talk offline with you about how to gain broader visibility.
Thank you. Thank you so much.
Thank you, Amy. I'll be happy to make a meme for you. That's the best way to get viral nowadays in this society. But jokes apart, thank you for being here. I'm Nerea. I'm from Spain. So happy that you're presenting for us. Hopefully we can have you in our country at some point. And I'm also part of iGEM EPIC. And my question is for the three of you. And at the beginning of this century, we've seen how reading DNA was a very expensive technology and took a lot of time. And now that has decreased dramatically. But now the barrier or the obstacle is, for example, mixing stem cell technology and gene therapy. That's something that could have a great therapeutic impact, but it still takes a long time and it's very expensive. Do you think it will have a similar pathway to the DNA reading technology? Could we ever achieve a cost and a time that make it available for widely use?
I'll tell you, I'll take a crack at that. I think that gene therapy is very closely related, some might say identical, to the methodology that was used for the BioNTech, Pfizer, and Moderna vaccines. So it's basically a gene, or maybe a couple of genes in the future, in a molecular surrounding that allows it to be delivered efficiently to cells in the body. So that is, those companies are interested in gene therapy. That is an example of it. It is a special case because you have the immune system amplifying whatever you deliver. But there may be lots of other special cases for gene therapy. So it's also a special case because it has such a large market that it can be inexpensive. So it's $15 a dose. So in stark contrast to rare diseases where you might have millions of dollars per dose. So one of the critical things is it's not just bringing the technology to the point where it could be cheap, it's having the market size that you can actually make it cheap. And so I think gene therapy is being subject to exactly the same exponential as the other reading and writing technologies.
I would just ask here, George, also if the virus-free cell line as a biomanufacturing platform will also drive down the cost of creating gene and cellular therapies, and that being one of really the ideal outcomes of that really tremendous project that we're trying to launch out of George's lab by driving down the cost of creating gene therapies, making them more stable and robust and cost effective to be able to be disseminated more broadly internationally and to third world countries. And maybe if you want, could touch upon that.
Yeah, absolutely. I mean, the ultra-safe cell and many other projects in GP Wright are likely to reduce the cost of cell-based manufacturing. And if we can develop an ultra-safe cell that is universal, meaning that it can be transplanted into anybody, then that also opens up the door to cell-based therapies that could have a faster route through the FDA because it's using the same cells with very small changes. Just like the influenza vaccines each year are relatively minor variations on previous ones, and they get approved more swiftly. So yeah, we're very excited about GP Wright's potential impact on equitable access around the world to these new technologies for agriculture, for manufacturing, and for healthcare.
Thank you. Hi, I'm Marcel from Germany. I'm also part of the iGEM team, and thanks for taking the time. So in your presentations, you've all painted some sort of picture of the status quo of genetic engineering and what we're able to do in terms of writing DNA. So my question is about the systems of increased complexity. When we think about moving from bacterial cells and mammalian cells to tissues or organ systems, even organisms, do you think the techniques to be able to program on this level will arrive with the same pace, or are they equally close on the horizon? And if not, what are some of the barriers that we will be facing? Thank you.
Well, in a way, we've already demonstrated a pretty good proof of concept with the pigs. Those are heavily edited, not just writing our names in their DNA, but edited for 42 different functional components, all of which have been checked. The functionality works, the new functions work, and the pigs are breeding healthy generations, and the organs are being used in preclinical trials in 3 hospitals. So I think that's evidence that we can deal with complex systems. It doesn't mean that every complex system is going to be easy, or even the pig xenotransplantation was easy. But there's some complex systems that are far beyond their understanding that we still manage to engineer. That's the sort of the difference between engineering and understanding is, and my favorite example is smallpox. We had not nary a clue about it, virology or immunology, nothing. But we nevertheless, the strategy that we used is at the beginning was very similar to the strategy that finally did eliminate smallpox and made it one of the first technologies that was truly equitably accessible to everybody in the world, which was the extinction of a major killer. So I think complexity should not intimidate us, but we should be cautious and careful, following FDA guidelines, making sure we have approval and widespread discussion. But it's amazing what engineering can do in spite of very highly complex systems.
Okay, thank you. I'm from the iGEM Arabia Mars team from Navarra, and this has been a very inspiring session, so we are very thankful for that. And we are from high school, so and we are working with transformated plants, through synthetic biology. And our question is if you think that maybe the genetically modified organism will be in a future part of our lives. Thank you.
Well, genetically modified organisms are already part of many people's lives. People that, for example, diabetics that need insulin, the GMOs are making insulin in bacteria, which is a very synthetic system. Use of genetically modified crops is pretty widespread. Most recently, golden rice is now approved for use. That could save 1 million lives per year. So it's not just slightly cheaper tomatoes, it is actually 1 million lives per year. And the American chestnut may be planted in the wild soon, as it was completely wiped out. Now it's being returned, being blight resistant. Those are just a few examples, and probably it will be many more.
So thank you so much. We've got still like 8 minutes left, and I'd love you to talk about going to a more abstract level. I really like that Genome Project Right is also focusing on values, so and ethics. So maybe you can talk a little bit about the importance of the heart together with the mind and technology to push forward our humanity in a good way. I am also very interested in education. I think we have an equity problem, at least barrier. Not all over the world we can access to universities that where you're teaching. So how about sharing something about online education for synthetic biology from high school up to starting up companies? Thank you.
So one example that we've had, Amy can give additional information. One example that we have a lot of experience with, almost 15 years, is pged.org. It's aimed at slightly broader than just synthetic biology, but it does aim at everything from high school. There's actually high school learning materials online, so the teachers can pick them up and use them immediately. It goes up through congressional briefings in the United States, a little bit internationally, and then working with Hollywood screenwriters and other authors who are interested in getting the science, the cutting edge science, and getting it right. So those are examples of things we have done in kind of the broader fields of genetics, reading and writing. But we'll have some that are specific for GP Wright. We've already mentioned our dedication to equitable access by bringing down the costs of genome reading and writing by about 10 million fold. That's a start, but that's not sufficient. The education is a key component. If the price is low and nobody knows about it, that's as if we hadn't done it. So, Amy, did you have, or Andrew, other comments?
One other comment is it's not just about the cost of reading and writing DNA. There's the whole, all the facilities required for doing bioengineering. Labs are expensive to set up and to operate. And one of the things that really has been exciting me is the appearance of cloud labs, like cloud computing, over the last decade. They're not widely available yet. They're not widely dispersed. There's only a handful of them that are out and operating, but already there's a consortium of these biological foundries that are really highly automated labs that can be programmed for doing research and development work. And I think these are going to improve access for many people, particularly in developing nations where the infrastructure just doesn't exist yet, that will allow them to kind of leapfrog into bioengineering without having to build all the necessary facilities and infrastructure, and in many cases, even the regulatory processes, because they can operate in essentially a closed system. And because I believe these tools for this type of engineering are going to get faster, cheaper, better, I believe that we're going to start seeing biofoundries in a box. We're already seeing some of this with Codex DNA and a remarkable device they make called the BioXP that takes many of these foundry operations and lab operations and puts them in a desktop machine that's reasonably affordable. So I think we'll continue to see advancements quickly.
Thank you, George and Andrew. Yeah, and I would just
on values and ethics.
Sorry, yes, and on ethics, I was just going to say that one of our interdisciplinary working groups is an ELSI group, Ethical, Legal, and Social Implications. And I invite you to join the conversation. Please do reach out if you want to join that working group or participate in the conversation. Barbara Evans from our group is going to be speaking at our upcoming conference over October 21st to 22nd, and the agenda is being made available. So if you're interested in joining that conference, you can certainly hear her speak. We'll also have a biosecurity and ethics workshop during the meeting. So if you're interested in joining and participating in that conversation, we do welcome you. And then I would just add that the CAD is really meant to be a democratized tool for really broad accessibility to students. So it's going to be open access and no fees associated for academia for students, and you don't need a computer science background. So it's really advancing that sort of design phase of genome engineering and making that first step, that first critical step, really broadly available for experimentation on the CAD platform. And there also, I would just add that, the libraries that we hope to build out for rare genetic diseases will be able to serve as a catalyst for really, advancing gene and cellular therapies and making them much more cost effective and sort of just advancing those pipelines. So we're hoping that the platform serves a very useful tool broadly for society.
Okay, so we're coming to the last 4 minutes. I'll ask you a very short question, and I leave you with the final remarks, and then we will close. We are in Pamplona. This is Biospain. This is organized by the Spanish Biotechnology Companies Association, and also Sodena, which is a public investment fund from the government of Navarra. So my question for Amy is how the biotechnology companies in Spain could join your initiative, the one you're leading with the Industry Advisory Board. I want Andrew to also tell us what to do. We were infected by his passion when he was here. The kids that you met, now they are leaving us, and they are not kids any longer, and they are getting their degrees, and very soon they will be changing our world. So maybe you have a message for them. And for Dr. Church, I want you to advise how we can play a role for the world from Navarra. So it's big questions, but you have only 1 minute each. Amy, we can go in the same order. Amy, Andrew, and George.
Thank you, Ano. So yes, please do contact me, and I can put my contact information in the chat here. That's probably the best way to really find out about how specifically to be engaged on the Industrial Advisory Board. It depends exactly where you could tap in, and it's a longer discussion, but we certainly invite industry from Spain and beyond to participate at the board level and figure out if there are possible extensibilities that we could build into the CAD to really drive users to your services and platforms if they make sense within the foundry and incubator that I described. So I'll leave it at that, but thank you.
Thank you so much, Amy. Andrew?
Thank you for having me today.
I see these technologies, the ability to engineer living things, as being crucial for humanity thriving. I think it's really come down to that. We see, we're very aware of climate change today. We're aware of many things happening in the world. It's young people that are going to drive change, and I believe in large part with these technologies. So I encourage young people to push the boundaries.
George.
So you asked, what your group, broadly speaking, can do. Yes, your community, as Andrew mentioned, the playing field is very level, and it gets leveled every few years because the technology is changing so radically. So one group, one community that invested heavily in technology X has to redo it with the new technology, and so everybody has an equal chance. So I think that there's tremendous opportunity there, and really there's a lot of sharing going on worldwide. So I think that should make it quite feasible.
Thank you. So
thank you.
Thank you so much. We had a great conversation with George Church, Amy Schwartz, Kane, and Andrew Hessel. We're happy to have you with us from wherever you are in the States and here at the Core of Pamplona, and looking forward to meet you again. And it was very inspiring, and thank you so much for energizing us and sharing your wisdom and knowledge with us. Goodbye. Thank you.